Oh, hello there. Welcome to Lichen Laboratories. I'm your lead scientist, Casey. It's super great to have you guys here. I was just about to dig into the case of a mysterious cancer of non human origin found in the lymph nodes and lungs of an immunocompromised patient. If you'd like to read the case yourself, I'll leave the article link in the description below. Otherwise, feel free to stick around and hear about it from me. This is a dwarf tapeworm, scientific name Hymenalepis nana. It is the most common tapeworm found in humans. There might be up to 75 million carriers worldwide. Most of these people are asymptomatic, meaning they're showing no signs of infection at all. Part of the reason this parasite is so widespread has to do with its life cycle. This tapeworm, unlike some other cestodes, has no need for an intermediate host, meaning the parasite can live its entire life cycle within the gut of a human, leading to a higher parasitic load, especially among those who are immunocompromised. Extraintestinal infections are not unheard of in tapeworms, but they're pretty rare with H. nana. Today, we will be discussing one such case where parasitic cells were found in the lungs and lymph nodes of a patient in Colombia. These cells were unusual due to the fact that they had characteristics similar to that of a malignancy. However, the cells were noted to be far too small to be of human origin. Now, neoplasms do naturally occur in invertebrates. However, up until this point, they'd never been documented in tapeworms. Our case begins in January 2013 in Medellin, Colombia, where a 41-year-old man presented to the hospital with fatigue, fever, cough, and weight loss over the course of several months. It is important to note that this patient was diagnosed with human immunodeficiency virus in 2006 and was currently non-adherent to his antiretroviral therapy. His CD4 positive T cell count was 24 cells per cubic millimeter. For reference, a normal CD4 positive T cell count is anywhere between 500 and 1400 cells per cubic millimeter. The cutoff for AIDS occurs when the cell count gets below 200, so he was solidly in the AIDS category. A stool sample from the patient showed eggs of Hymenalepis nana and the cyst of Blastocystis hominis, a protozoan gastrointestinal parasite. A CT scan was obtained, which showed nodules within the lungs between 4 and 4.4 centimeters in diameter. There were also nodules present on the liver and adrenal glands, as well as lymphadenopathy or lymph node swelling, in the cervical, mediastinal, and abdominal lymph nodes. A cervical lymph node was excised for biopsy, and a core biopsy of a lung nodule was also obtained. These were sent to the CDC for further examination. While diagnosis was pending, the patient was being treated with albendazole, a broad-spectrum antiparasitic drug, as well as being reinstated on his antiretroviral therapy. The disease, however, continued to progress over the next few months, and in April of 2013, a second cervical lymph node biopsy was obtained and also sent to the CDC for testing. The lymph node biopsies showed grossly abnormal, solid, and nodular masses made up of small, atypical cells with scant cytoplasm and very prominent nucleoli. These cells were only 5 to 6 micrometers in diameter, which made them slightly smaller than a human red blood cell. So they were very small compared to the surrounding lymphoid and lung tissue. The nucleus itself took up a good amount of the space, being 2 to 3 micrometers in diameter. These abnormal cells disrupted the normal architecture of the tissue around it by developing into crowded clusters of the small, abnormal cells. Syncytia formed on the exterior of these clusters. meaning that the cells themselves fused into large multinucleated masses with one large cytoplasm. Other than these syncytia, however, there were no further differentiating features present in morphology. Immunohistochemical staining, aka IHC, showed an absence of human cytokeratin and vimentin, which are often present in cancer cells. The stains were also negative for any free living amoebas. These features led to quite the diagnostic mystery. The cells showed many features of traditional malignancy, including their tendency to invade normal tissue, a distortive growth pattern, the cells being monomorphic or from a singular cell lineage, and the cells showing characteristics of immature stem cells, such as the large nucleus and prominent nucleolus. However, as previously stated, these cells were small enough to make the possibility of infection much more likely. Tapeworm infection was not initially considered due to the masses lacking the typical cellular architecture of a cestode. Instead, a plasmodial slime mold was considered, as well as other free-living amoebas. A cell culture for amoebas and slime molds was attempted, but no growth was noted after 4 weeks. A PCR was then run on the abnormal cells using genes that encoded ribosomal RNA. First, a pan-fungal and slime mold-specific PCR were run, with no matches. Later, a cestode and then hymenelepidid species-specific PCR were run on DNA extracted from the abnormal cells. The identification of Hymenelepis nana was unexpected, but it was a 99% match on PCR. However, there was an additional feature to these DNA segments that was found upon further study. 3 single nucleotide insertions occurred over the span of 12 base pairs within a region of highly conserved information, possibly the reason for the morphological changes. The patient during this time, however, continued to decline. His cervical lymph nodes continued to swell, and eventually the combination of medications caused renal failure. He declined hemodialysis, and palliative care was started in May of 2013. A molecular diagnosis was provided to the patient only 72 hours before his death, and specific treatment was not attempted. Before his death, the patient provided written consent for the studies to be performed and the research to be published. So, why didn't anti-parasitic treatment work for this patient? If he had an infection, even if it was similar to malignancy, it should have had some effect, right? Well, the cestode stem cells were cultured and then treated with albendazole, the anti-parasitic the patient had been treated with previously, and the cells seemed to have a resistance to the drug in their immature form. So this would have been a barrier difficult to overcome even if treatment had been attempted. Now, this case study teaches us a few things about Hymenelepis nana and cestodes as a whole. For instance, this and the studies involving immunocompromised mice suggest that normal tapeworm development possibly requires a normal host immune response in order to properly differentiate cells. Atypical development in other species of tapeworm has been seen in other immunocompromised humans, as well as in other mammals such as orangutans and cats. However, in these cases, the cellular architecture always remained recognizable as that of a tapeworm. This case is novel, and nothing quite like this has been documented before or since, making this perhaps the rarest malignancy in the world, with only one case diagnosed in history. It certainly shows the importance of research on immunocompromised animals and how they react with infective agents. I hope you enjoyed this case study. As I said in the beginning, if you want to read the publication yourself, it is linked in the description below. Be sure to subscribe if you'd be interested in more content like this. Thank you, everyone, and I'll see you next time. Bye-bye!